Hi, this is Justice Pivito giving my graduate presentation for Dr. Kuknor's molecular biology class over epigenetics, ethanol exposure, and alcohol use disorder. So alcohol use disorder is estimated to cause 100,000 deaths per year in the US. It is a condition from which 1 in 12 Americans suffer, according to the AAC. Given these facts, it is clearly a widespread and significant issue. Adding to this issue is that some individuals can be more genetically predisposed to developing this condition. There's long been a common understanding that some individuals are more likely to develop alcohol use disorder, but only relatively recently have the links between heredity and a predisposition for alcohol use disorder been found using genetic techniques. While genetics and heredity play a large role, and studies have shown that alcohol use disorder can run in families, statistically, one's likelihood of developing alcohol use disorder strictly from their genetics, that's only half the risk. There are many human factors to consider, but a largely ignored aspect regarding the development of alcohol use disorder is the environmental effect on one's gene expression, i.e., epigenetics. So epigenetics is a fairly new field, and it's super interesting. It's a field that I have been interested in for a while. So epigenetics is defined as a phenotypic change without a genotypic change. This is changing how we see the human genome and has potentially huge implications for our understanding of the wider field of genetics. The classical view that one's development or propensity for a disease could be largely predicted from their genome is fast changing. Epigenetic changes on DNA and histones such as methylation, acetylation, phosphorylation, and many more can change gene expression. This field has wide applications from genetic engineering to hereditary disease. Epigenetics may have a significant impact on human development and pathology, and further study could facilitate a broader understanding of human health and disease. Yet experiments involving epigenetics are nascent. Because the epigenome is so different from our classical understanding of genetics, experimentation can be difficult. Nevertheless, there are studies researching epigenetics and alcohol use disorder. An alcohol research current reviews paper has found evidence of chromatin remodeling in the amygdala following histone modifications. DNA methylation is also active in regulating gene expression and is thought to play a role in the development of alcohol use disorder, according to a 2012 paper by Starkman et al. This paper pulls from many studies with numerous experiments using similar methods. It has been found that exposure to ethanol can inhibit histone deacetylase, or HDAC, and DNA methyltransferase, or DNMT, activities. Acetylation of histones has been shown to be regulated by HDACs and histone acetyltransferases, or HATs, which remove and add acetyl groups from histone respectively. When acetyl groups are added, the binding between the negatively charged DNA and the positively charged histone are loosened, resulting in what would be called euchromatin, which is more likely to be expressed. So less acetyl groups causes increased interaction between the N-termini of histones and the negatively charged phosphate of DNA. So this tightens the DNA structure and lessens a nucleosome's likelihood of being transcribed. So when HDACs are inhibited or HATs promoted, then DNA is more likely to be expressed. Methylation of histones generally has the opposite effect. So when DNA itself undergoes methylation, so this isn't the histone, this is the DNA itself, another epigenetic process where methyl groups are added to the DNA structure, often on the fifth carbon of a cysteine ring. This represses gene expression due to steric and torsional interferences between enzymes and the new methyl. So DNMT enzymes regulate DNA methylation, and there are three types. DNA methyltransferase, again DNMT, is intimately involved in this process and is one of the main enzymes that adds methyl groups to DNA and helps silence genes. This is an important process in gene regulation. While research related to this has brought to light certain epigenetic effects caused by ethanol exposure, there are still more details to be discovered. The exact mechanism of acute ethanol exposure on enzymes such as HDAC and DNMT is still unclear. Insight into the exact mechanisms and processes could have medical benefits. How ethanol exposure affects other important DNA expression regulators may not have been studied either, and the literature could benefit from an expanded study. So, for example, histone demethylases and methylases are important regulatory factors that have not seen much study. The studies mentioned prior have shown evidence of changes in histone acetylation and DNA methylation in response to alcohol exposure, but less study has been done on histone methylation and other epigenetic factors. It's actually only been shown this year, 2021, that a histone demethylase, or KDM6B, also known as JMJD3, is dysregulated in alcohol-dependent rodents in the experiment, as they were called, and human brains. This was a study by Johnstone et al., 2021. So further study could benefit from a focus on histone methylation and demethylation. So as of present, studies in this field have merely laid the groundwork for true understanding of the epigenetic effect on alcohol use disorder. So we aim to find a better understanding of the enzymatic activity in response to ethanol exposure by striving to link the epigenetic modifications already studied further to specific levels of alcohol consumption. So factors only recently studied in detail before, such as DNA and histone demethylases and HMTs, will also be evaluated quantitatively. However, understanding the intricacies of epigenetic biochemical modifications that have not been studied previously is not really the intention of the study. We will measure and focus specifically on histone methyltransferases, HMTs, histone demethylases, HDMs, histone acetyltransferases, or HATs, histone deacetylases, HDAC, and DNA methyltransferase, DNMT levels. This study will aim to better understand epigenetic modifications, principally on histones, but also importantly on the methylation and modification of DNA itself. So there would of course be many other proteins and enzymes in the body that we can secondarily measure during this experiment. CREB, or CREB, a protein which is linked to long-term memory and addiction, also helps regulate the expression of genes tied to alcohol use disorder. Another is BDNF and neuropeptide Y, or NPY. Lower levels of NPY mRNA and protein have been found in so-called alcohol-preferring rats in previous studies when compared to alcohol non-preferring rats. This suggests a link between NPY and alcohol drinking behaviors. It was also found that acute ethanol exposure increased the expression of ARC and BDNF in the amygdala of alcohol-preferring but not non-preferring rats, as well as increasing CREB phosphorylation levels. Furthermore, alcohol-preferring rats that have never been exposed to alcohol showed less expression of other CREB target genes, such as BDNF and ARC proteins, in the medial and central amygdala compared with non-preferring rats. We will also measure levels of these proteins and enzymes and at what levels of alcohol exposure they are found. So that being CREB, BDNF, neuropeptide Y, and ARC. Behavioral data will also be collected. On to our general experiment. We propose using non-human primates as subject animals, specifically macaques. They're more closely related to humans and will help to gain a more accurate understanding of human alcohol use disorder. By obtaining behavioral data and comparing it to our more quantitative molecular data, we will gain novel insight only seen in some prior studies using rodents. Measuring specific enzymatic amounts and activity, we will be able to discern the amount to which epigenetics specifically affects behavior. However, it should be noted that the primate's hereditary data will not be as effective as a rodent model as a result of longer gestation periods and life cycles, so generational study will be very limited compared to a rodent. It's already known that the classical heredity affects one's likelihood of developing alcohol use disorder, but data on epigenetic changes being passed from one generation to another is sparse. So, if the study can be continued for several years, we will be able to gather data and see to what extent epigenetic factors specifically change a subject's likelihood of developing alcohol use disorder and certain changes in the epigenome, genome, and behavior. This study will also use a hereditary control group to compare the data to. Rodents have widely been used in previous studies, but they're not perfect models. So they metabolize alcohol much faster than primates and have many physiological and behavioral differences to humans. So the crab-eating macaque, Macaca fascicularis, and the rhesus macaque, or Macaca mulatta, have often been used in research on human pathogenicity. The precedent in current literature and the availability will make these species more practical and cost-effective than other primates, such as the chimpanzee. While chimpanzees and other apes like bonobos are more closely related to humans, they have a much longer gestation period and longer lifespan. Additionally, apes are harder to get simply, and they're more expensive. So I'm going to go ahead and go with macaques. Macaques have a gestation period of roughly 166 days. They mature around 4 to 6 years, depending on the sex or the species, and they have a lifespan of around 30 years. For studying epigenetics and heredity, they're kind of a middle ground between rodents and apes for the purposes of expediency and genetic and behavioral similarity. So many traits are more likely to be passed through either the paternal line or the maternal line. We all know that. Some evidence indicates that epigenetic factors can have a similar paternal or maternal inheritance. The germline itself can be epigenetically altered, which is very significant. Some studies suggest that certain paternal exposures can have consequences for offspring development, even down to the third generation. Paternal epigenetic changes, such as malnutrition, have also been shown to have effects on ensuing generations and can also affect maternal behavior, such as increased infant care and mate selection. That's from a study from 2018. So sex-related epigenetic inheritance is not well understood. We plan to measure our non-genomic changes in parent generations and observe how this affects offspring, taking into account sex, behavioral changes, and epigenetic abnormalities. At this point, it's probably clear this is a pretty multi-purpose study. It's going to be behavioral, qualitative, quantitative, focus on generational changes, compare paternal, maternal, epigenetic lines. So this is a multifaceted study, borders on becoming multiple studies. However, because we will be using non-human primates, this study will take much longer, and the researchers will have more time to record and interpret data as the experiment goes on. Additionally, the cost of obtaining and raising non-human primates is not insignificant, and so a multifaceted study will be an efficient use of resources and time. Studying multiple aspects of alcohol exposure, epigenetics, and epigenetic inheritance will yield the most results for the cost of the experimentation. So many studies have used mice and simply measured enzyme production after intraperitoneal alcohol injection. This is because mice and other rodents will not willingly drink large amounts of alcohol. Another reason why we're using rhesus monkeys here. We propose using a different method of alcohol administration. The alcohol administration approach in this experiment will be oral. Previous related primate alcohol studies have used ex vivo techniques, but we'll be gathering behavioral data, enzymatic data through blood testing, and we'll be obtaining postmortem tissue data after a subject macaque dies. We will have a group of macaques and regularly give them the option to consume alcohol at varying amounts by group and record their behavior, and then take regular tests. So there will be 4 groups, one with no alcohol consumption, that's the control, one with very light alcohol consumption, one group with moderate alcohol consumption, and one with heavy alcohol consumption. We'll go into the details on those levels later. So our general measurements and animal treatment conditions will be largely based on a previous study that was approved by the Institutional Animal Care and Use Committee at West Forest University Health Sciences and conform to the National Institute of Health's guideline for the care and use of laboratory animals. So this was in research by Davenport et al. 2013, listed in references. So group 1 will consume no alcohol. Group 2 will consume about a half to 1 gram per kilogram, which is the low amount. Group 3, with the moderate amount, will consume around 2 grams per kilogram, while group 4 will drink upwards of 3 to 4 gram per kilogram, which is kind of a lot. This is, again, similar to some previous experiments. So the primates will then breed within their experimental group. There will be offspring where the mother drinks regularly and the father does not, and vice versa. This will allow us to gather data on the previously observed non-genomic paternally and maternally influenced changes on child development and alcohol use disorder traits. The levels of our enzyme of interest will be measured and compared to the primate's behavior and their parents' data. By measuring the enzyme production levels of DNMT, HDAC, HMT, HDM, HAT or HAT, and also CREB, BDNF, and NPY in the amygdala and the blood, we will be able to link the observed behavioral and phenotypic results to the biochemical differences in the control population and conclude to what extent epigenetics were at play. Histological, genomic, and proteomic assay techniques will be performed on brain tissue from regions associated with the items of interest, along with blood tests. So, regarding observing proteins in the amygdala, though, a detailed craniotomy procedure must be performed at death, as in prior studies by Davenport et al. 2013. As done by past experiments by Cheng et al. In 2004 and 2010, each animal would be perfused for about a minute and a half with ice-cold ACSF buffer containing a whole cocktail of mixes that is detailed in the paper. So the use of a large bore cannula will allow the brain to be perfused with approximately 1.5 to 2 liters of cold ACSF. So during the perfusion, additional tissue will be collected, again, as in Cheng et al. 2004 and 2010. For determining specific enzyme and protein prevalence, we will compare the amount measured in the control, or group 1, to each of the other 3 groups and determine if ethanol alcohol exposure has a statistically significant effect on the amount measured for each group. If these amounts match previous studies, e.g. lower levels of HDAC and DNA methyltransferase, or DNMT, activities for groups exposed to alcohol more, then we can corroborate those previous findings and contribute more quantitative data to the current literature. If the levels are different than previously found, then the experiment might have run into some issues, or there could simply be more at work in this field than previously thought. Although, given the levels of research done in this specific way, we expect to find somewhat similar results for specific protein and enzymatic levels corresponding to low or high levels of ethanol and alcohol exposure. However, with our varying levels of group exposure, we hope to find at what point these epigenetics effects become pronounced. That's why we're kind of leveling, stratifying these groups per level of alcohol. So this will contribute to a wider understanding and potentially aid in human health by measuring out a specific safe amount to consume before non-genomic changes become particularly hazardous. So regarding behavior from parent to offspring, We hope that we will be able to discern the level of alcohol use traits that have been passed on and how much non-genetic traits affected the likelihood of alcohol use disorder traits. With our groups of varying alcohol consumption, we will be able to compare, hopefully by the third generation if the experiment is allowed to run that long, which groups have been affected. For example, if a family line started with no alcohol use disorder traits, and through alcohol exposure they began to gain these traits, and then this is corroborated by our data, measured qualitatively or, maybe showing dependence on alcohol, and quantitatively by less production of certain peptides like NPY, then that would be data of interest that we would note. Lastly, if there is a significant statistical difference in alcohol use disorder traits between maternal and paternal lines, this would give further evidence to the notion that epigenetic germline changes can have different effects depending on the sex of the parent. So that is also something of interest. And here are the references. Quite a few, but yeah, thank you so much for listening.